Well, good afternoon. Thank you for coming back. Those of you that did and got fed, not all of us did get fed, so we brought back boxes of food with us. I'm going to now introduce and welcome our next speaker, Tom Whitham, the Regents professor from NAU. And with that, I think we'll just keep moving along here. So, thanks, Tom.
Good afternoon, everyone. I'd like to thank Lauren for inviting me. And today what I'd like to talk about is climate change as being both an ecological event and perhaps even more importantly, an evolutionary event. As plants become maladapted to their local environments, I think it's going to force us into using assisted migration and genetics-based ecosystem engineering. I don't necessarily want to do that, but I think the issue will be forced upon us. The photograph you see here is a heat map of my own body, and all organisms, or most organisms at least, are sensitive to temperature, and temperature has been a selection pressure that they've evolved to. And so anything that affects temperature, such as climate change, has very high potential to be an agent of selection and to select for different genotypes and resulting then in evolution. And so one of the primary tools that we're using to examine climate change as a selection pressure is the Southwest Experimental Garden Array. This is an array of 10 common gardens on an elevational gradient, and new common gardens are being added to this all the time. And the broader the array, the greater the resolution that you're finding, just like the radio antennas in New Mexico, giving you greater resolution in radio astronomy. And so here's one of the sites out at the Arboretum there at Flagstaff. And the reason this is a facility that's open to anybody from people studying microbes to vertebrates and grasses to trees. And the important aspect of this is that it allows you to examine how locally adapted plants really are. Okay, and so this is really an important issue for climate change research, because the more locally adapted you are to the local environment, the more locally maladapted you will become with climate change. And so it's really important to understand the level of local adaptation. Okay, so this common garden array is on an elevational gradient, and so by planting seeds or clones of plants along the gradient, by putting them at a lower elevation, you can expose them to very predictably hotter and drier conditions as a surrogate for climate change. And then by watering those plants, you can eliminate water as a variable and just study the effects of temperature by itself. I should also mention that these gardens are on lands with long-term leases from the National Park Service, BLM, BOR, the Nature Conservancy, Game and Fish, Grand Canyon Trust, Abbott Ranches, the Arboretum. And the reason that they are giving us long-term leases on this land is that they want to know the basic research and its implications and solutions to improve land management on their adjacent lands. Okay, so I'll be talking about cottonwoods as a model system today because it's the first tree to have its genome sequenced. The genetics resources to understand climate change are really great with this species. And in our studies using common gardens and studies in the wild, we now know that there are at least 3 different ecotypes of Fremont cottonwood. And there's Utah High Plateau, and so it's higher elevation, cold adapted populations. Then there's the Central Valley, California ecotype, hotter environment with a different precipitation regime than the Sonora Desert ecotype. And these are all genetically differentiated. We could call them subspecies. And this shows some of the genetics data, which has unfortunately been cut off here, but it shows that they are so genetically differentiated, we might even go so far as to call them different species. Okay, so an important issue in climate change modeling of species distribution, right now the models really rarely, if ever, take into account genetics. And so all of these ecotypes just get pooled into one modeling effort. And so this is some modeling by Akita in a global change biology paper. And what we're doing is modeling ecotypes as if they were different species, because they're so genetically differentiated, it doesn't make sense to pool them. And so this looks at the California ecotype, its projections from 2050 to 2080 and current. And you can see that this ecotype, its longitude and latitude distribution is not projected to change at all, at least up into 2080. But then the Utah High Plateau ecotype, its distribution is projected to shift quite a bit, and you can see the confidence limits on this. And then the Sonora Desert ecotype is projected to shift even more. And so what you can see here is that they're very different from one another as well. And so again, it doesn't make sense to pool these genetically differentiated ecotypes or populations. We really need to look at them separately. And a justification for doing this is shown in this slide, where we use genetics-based models and compared them with no genetics-based models. And then we examine how good they are at predicting ecoregion test points. And what we found were that the genetics-based models were about, were about 12 times better on average in predicting test points than were models that didn't have genetics incorporated in them at all. And so here you can see for each of the three ecotypes, this shows the genetics-based model and the no genetics model, and predictability gets higher as you go down. And so this is just another justification of why this kind of modeling, incorporating genetics, should be a productive route to go. Okay, so even within an ecoregion, there's tremendous genetic variation that needs to be considered. And so here is a common garden approach to looking at local adaptation within a single ecotype. And this is the Sonora Desert ecotype. And here we have each leaf represents a different population throughout the Sonora Desert ecoregion. And then the leaves with a circle around them represent common gardens. And so what we did is we collected plants from each of these populations, we propagated them in the greenhouse, and then planted them out reciprocally in each of these common gardens. And so 16 populations reciprocally planted in each of these gardens. This garden is the upper elevational naturally occurring distribution of Fremont cottonwood. It's at Canyonlands, and its mean annual temperature is about 10.7 degrees centigrade. The lowest elevation is double that temperature at about 22.8 degrees centigrade on BLM land near sea level. And then there's an intermediate elevation common garden at Arizona Game and Fish Lands which are embedded in Agua Fria National Monument. Okay, so everything is reciprocally planted here, and so we can really quantify pretty precisely how locally adapted these plants are. This next slide shows you one of the common gardens. This is the one at the intermediate elevation site at Agua Fria, and it's bounded by Agua Fria Creek on this side and Indian Creek on this side. And here we have an L-shaped common garden along the creek, and there's about 6,000 or 5,000 trees in this garden, again with 16 populations represented. Okay, so here's some data from these 3 different common gardens. Here we have the high elevation common garden where it's the coolest of the sites, and it has a relatively short growing season. And here you can see of the 16 populations, these dark blue ones represent ones from colder sites. And the colder sites survival is about 2 times greater than the ones from low desert moved up to higher elevation. And so this shows the level of local adaptation. And you see just a reverse pattern at the hot garden with the long growing season down near sea level in the southwestern part of the state. The blue dots represent ones from hot sites planted in a hot garden. And you can see that their survival is about 2.5 times greater than the high elevation populations moved down into the desert. And we see the same sort of thing occurring with the intermediate site. At the intermediate site, the populations from intermediate elevations significantly outperform the ones from cold sites moved into intermediate and hot sites moved into intermediate. And so what this is saying is that they're all locally adapted, and it turns out that for about every 1,000 feet in elevation that you increase, you get a genetically differentiated populations that are locally adapted. And so this is pretty fine scale local adaptation. The good news on this is that plants have evolved to deal with the environment, biotic and abiotic environment. The bad news is that with climate change, we're pulling the rug out from underneath these plants, so what is locally adapted today or yesterday is no longer going to be adapted tomorrow because we're changing the environment faster than they generally can evolve to deal with it. Okay, so another really important thing about these common gardens is that they allow you to identify the genes and functional traits that are under selection. And so this is known as QST, FST analysis. And so this solid line here represents FST or the neutral markers. And QST looks at if you deviate from this line significantly, you're seeing traits that are under natural selection or artificial selection. It wouldn't be the case with climate change. And so then bud set, bud flush, height, diameter, specific leaf area, these are all functional traits that are under selection and have resulted in the divergence of these populations from the natural distribution at the lowest elevations to the highest elevations. And so this is really important because it allows ecologists, evolutionary biologists, climate change modelers to identify the specific traits that are going to be selected for or against under different environmental regimes. And the selection, you can see that these functional traits, these are for the 3 different gardens, you can see that they change. And so what's necessarily under selection in one garden will be not selected in another garden. So important to keep this in mind, but this is a really powerful tool because it allows you to really take climate change to a genetic level to really understand what's going to be under selection. Okay, so the first part of my talk was to emphasize that things are locally adapted. And that's bad news in terms of climate change, because the more locally adapted you are today, the more maladapted you will be tomorrow. Okay, so the second part of my talk is I want to talk about how the genetic differences in these functional traits largely define the associated community and ecosystem processes. Okay, and so, and the importance of understanding this is that with climate change, anything that affects the plant is going to cascade to affect the beavers, the birds, the insects, the fungi, the soil microbes. Okay, and so we can show that beavers are very sensitive to the genotypes of the trees. They just don't randomly fell trees. They are very selective in what they're looking for in those trees. Same thing for the birds, the insects, the fungi, and the microbes. Okay, so any genetic change that occurs due to climate change on a plant is going to cascade to all of these other organisms. Okay, so just to give you one example of this, this is a study by Zacchaeus Thompson, and these are insect emergence traps placed in a stream. And so what Zacchaeus did was he collected leaf litter from individual tree genotypes, and then he put the genotype, the leaf litter of an individual tree genotype and an individual insect emergence trap in the stream. And then at the top of those traps, he collected the macroinvertebrates that emerged from after feeding on the leaf litter that actually emerged. And so you can see it replicated all the way down the stream there. Okay, so then he looked at the effect of tree genotype on the macroinvertebrate community. And so basically what this is doing is looking at the genetic footprint of a tree in a terrestrial system as those leaves fall into an aquatic system and support the macroinvertebrates. And so what we have here are 7 different tree genotypes. This is an ordination, a community analysis ordination with confidence limits. And so here are the communities from one genotype, communities from another genotype, and so on. And so 2 tree genotypes that overlap have very similar macroinvertebrate communities. Those that don't overlap, like these 2, have very different macroinvertebrate communities. And so just by knowing the genotype of the tree, he could predict 45% of the variation in the macroinvertebrate community. And so this says genotype is very important. And this is a very important argument of why you want to conserve genetic diversity, because if different tree genotypes support different communities, the greater the genetic diversity in the plants, the greater the diversity in the associated communities. And we've done this same sort of experiment with lichens. We've done it with insects on the leaves, pathogens on the leaves, microbes in the soil, and they all show that the genetic component to community structure is between 40 and 70%, depending upon which community you're looking at. So there's a strong genetic component to community structure, and which genotypes you plant or are out there naturally, they're going to play a major role in defining those communities. And they represent heritable traits passed from one tree generation to the next tree generation. Okay, so just to give you a little bit of a feeling about these cascading effects of genes to ecosystems, here's a system we've studied for quite some time, looking at map traits for resistance to an insect, a common insect, a gall-forming aphid. And here are 3 different genes that are associated with resistance to this gall-forming aphid. And basically, whether a tree has 1, 2, or 3 of all of these resistance traits will determine just how resistant it is. It's a quantitative trait. And so here is the traditional phenotype of resistance in the tree population. And you can see that some trees are basically death chronicles because 0 aphid survival. And then other trees are basically aphid candy, 90% aphid survival. And this is the distribution in the population. So that's the traditional phenotype. But these phenotypes have community and ecosystem phenotypes as well. And so here's a study by Laura Dixon showing that whether or not the trees are resistant or susceptible affects the birds that feed on the aphids, the fungi that feed on the honeydew, the insects that feed on the aphids, spider inquilins. There are about 60 species in that analysis. And so the genes here have an extended phenotype on this community. It also affects community stability. The presence of aphids results in a stable community year after year after year. If you don't have aphids, the communities fluctuate a great deal. And so this is a study by RP showing community turnover or community stability across years. And then this is a tritrophic level interaction study by Joe Bailey showing how the presence of aphids affects the foraging birds like black-capped chickadees that feed on the aphids and other species that would use, vertebrate species that would use this aphid. So these are community phenotypes of these genes. And then there are ecosystem phenotypes. And so Jen Schweitzer did a study looking at litter decomposition. And she found that aphids trigger an induced response in the tree in which, in the presence of the aphid, the tree shows a lot of condensed tannins into those leaves. And so and the leaves with the aphid have higher condensed tannins than leaves without the aphid. And so when those leaves fall to the ground, the condensed tannins inhibit decomposition. And so now you can show another community phenotype of these genes and effects on decomposition rates. And so these are the kind of links that you should be able to detect and map for individual genes at the community and ecosystem level. Okay, so now that we've sequenced the genomes of various plants, cottonwood was the first, and so the genetics resources are really among the greatest of any organism. There's a huge effort to map functional traits onto the genomes of various organisms. And so here's a genetic map that Scott Woolbright in our group first published in 2008. And on this genetic linkage map for Fremont cottonwood, we're now mapping functional traits. And this will be a 100+ year effort. I mean, this is just going to be ongoing, and more things are going to be placed on these maps. And so this looks at the so this looks at two chromosomes that have two phytochemical traits of salicortin and condensed tannins. These, as phytochemistry, they have a big effect on aphids, they have a big effect on beavers, and so these are very important secondary compounds in these leaves. Another functional trait is phenology, spring leaf flush, fall bud set. We can show that in the cottonwoods that phenology accounts for about 70% of the variation in productivity. So hugely important traits. They trigger hormonal systems in mammals that feed on the plants. They have similar effects on insects. Very important trait. This looks at functional traits for architecture. About 70% of the variation in the architecture of a tree is genetically based, and birds really key in on this because a sharp branch angle is preferred by, say, robins, and narrow branch angle is preferred like northern oriole. And so magpies, most birds have, if they make a nest, they're going to relate to architecture. Architecture also influences sink-source relationships of how resources are translocated around in the plant, and that affects all sorts of organisms, from pathogens to insects. And then here's the one I talked about, the resistance to pemphigus beta, this gall-forming aphid, and its cascading. So for each of these things, I can give you the cascading effects that have community and ecosystem phenotypes. And so we always read anything that affects the genes of a foundation plant like a cottonwood or a spruce. Take your favorite plant, it's is likely going to have these effects. Okay, so you might ask the question, why do different trees support different communities? And the reason for that is that there are a lot of functional traits. And so here are functional traits that we've already quantified from specific leaf area, photosynthetic rate, phenology, bark thickness, frost tolerance, heat tolerance, growth, reproduction, ontogeny, induction, phytochemistry, resistance. So every plant is going to have all of these traits being expressed. But due to assortment during sexual reproduction, some trees are going to have a lot or none of all of these combined traits. And that means that every individual in the population is going to be likely, unless it's a clone, is going to be somewhat different. And that's why you get different genotypes of trees supporting different communities. And so then this shows all the extended community phenotypes that we see from these functional traits. Canopy arthropods, decomposing fungi, interaction networks, avian nesting. All of these are community level traits that are heritable, and then you can take it down to the ecosystem level of carbon cycling, carbon pools, biodiversity, stability, trophic structure. They all have a genetic component that can be tied back to those functional traits at the individual and population level. So you can actually quantify heritability of community and ecosystem phenotypes. Okay, so the next point in this section I want to make is that communities are far more linked than we previously envisioned. Okay, and so here's a study in a common garden by Lamb et al. In Journal of Ecology, and this is a community interaction network. And this looks at community genetic correlations among very different communities. And basically what a community genetic correlation is that, again, it's cut off here at the bottom, but the changes in composition of one community among plant genotypes are mirrored by changes in the composition of other communities. And so what that means, if you change the genetics of the tree and you affect one community, it will cascade to affect all of these other communities. And so here shows all of the possible combinations. The dark lines represent significant interactions. The light gray lines show possible interactions which weren't significant in our analysis. And so if you look at the insects on the leaf canopy, if you change the genotype of the tree, you will change this community. And if you change this community, you will affect the ectomycorrhizal fungi in the roots, you will affect the lichens on the tree trunk, the fungal leaf pathogens, the twig endophytes, the soil fungi, and the soil bacteria. And so it is all interconnected. And so this is one of the first examples that I know of Darwin's entangled web that he first wrote about over 100 years ago. But here's the actual demonstration of it. Everything is really far more connected, just as he proposed it would be. And you can show it at the genetic level. Okay, so if you change the genetics of a tree due to climate change, it's not only going to affect that plant, it is going to cascade to affect all of the organisms that are dependent on that plant for their survival. Okay, so the third part of my talk is that climate change then creates a mismatch of once locally adapted plants and the new environment. And so this is going to then, the greater the mismatch, the lower the performance or eventual death of the plant, and that's going to cascade to affect the survival, biodiversity, and interactions such as mycorrhizal mutualism. Okay, so to give you, we need to be able to quantify the mismatch, because that's really key to understanding climate change and doing something about it before it's beyond our ability to do something. Okay, so So these hardiness zones are real important for plants because the USDA constructed them as a predictor for agronomists and silviculturists, horticulturists, because this is what they base where they're going to plant stuff. Okay, and so if you're dealing with an annual crop like corn and soybeans, it's still very important, but if you're dealing with a tree, a long-lived tree, what we plant today, we need to make sure it's going to survive 60 to 100 years. And so for many trees, if they're silviculture of it, if it's production forestry for wood, they don't harvest those things until at least 60 years in many cases. And so we need to plant not only for today, but we need to plant for tomorrow. And if the environment's constantly changing, then how do you match it up? And so it becomes really, really important to be able to project what foresters should plant. And of course, agronomists in the Midwest, they pay attention to this stuff because this affects yield. And a 10% change in yield for a farmer is the difference between making a profit and going bankrupt. So small changes here are a big deal. Okay, so we often talk about climate change as if it's something in the future, but I want to emphasize it is right now. Okay, and so this is an example where the mismatch has gotten so great that it's exceeded the physiological, ecological, genetic tolerances of the species. Okay, so this is the Bill Williams National Wildlife Refuge in southwestern Arizona near Lake Havasu. Okay, 10 years ago, this river was listed as one of the 10 most pristine rivers in the US. And one reason it was so important is that there are nearly 30 sensitive and listed species that are dependent upon this river for their survival. And these include things like butterflies to the southwestern willow flycatcher and the yellow-billed cuckoo and mammals. Okay, so what we've seen here is over a period of about 4 years of record drought, we have now lost, this photograph was taken a year ago, we've now lost at least 90% of the riparian vegetation. And you can just see it's kind of a sea of death, and you walk down into it, it's like pick up sticks, everything's pretty much dead. Okay, so the issue is not only important for the tree, it's really important for all of the organisms that this tree supports. And so this shows real-time climate change. And so if we adopt the mantra of most of our agencies of restoring locally, well, this is a really bad idea to restore locally here because the local stuff clearly can't hack what the environment has become today. And so now the question is, if we want to restore this and plant material that can survive the current environment and the future environment, where do we go to find the material that can actually survive this level of climate change? And what I'm talking about here is not, I'm not cherry picking here. I can go to rivers throughout the Southwest and find more examples of this that I wouldn't have seen before. And here's an example just north of the San Francisco Peaks in a pinyon juniper woodland. And so the dead trees are pinyons, the live trees are junipers, and so this is a codominant vegetation type, the most common vegetation type in Arizona, and this is virtually converted completely over to a juniper woodland. Well, there are about 1,000 species that live on pinyon, and so when you lose pinyon, say like pinyon jays have to have those pinyon seeds, and so this has a huge impact on things like pinyon jays. And so here's another example, and I think if you start looking at the ecosystems you're familiar with, particularly at the trailing edge of the distribution, where they're already at the edge of their distribution. This is going to become more common, and we're seeing vegetational shifts in distributions. Okay, so here we've also looked at the consequences of stress on the insect community. And so this is some work by Adrian Stone, and this just shows some of the common species that you find on pinyon. And so this is a study in 2010 by Adrian Stone, 155 arthropod species. This looks at species richness per tree and abundance of those species per tree. And then on the x-axis, which is cut off, this is a stress index. Okay, and the stress index is based upon branch dieback, number of needle cohorts, and radial trunk growth. Okay, so on the left side, we see trees that are under very low stress. On the right, we see trees that are under high stress. And so what you see here is that species richness declines to near zero on a highly stressed tree, and abundance of those species declines to near zero on a highly stressed tree. And so a lot of folks think that if you save the plant, you will save its associated community. And what this tells you is no, that's not necessarily true, because if you stress plants are almost worthless for most insects. And so if you stress this tree, you will lose almost all of the biodiversity that those trees support. And so we have to have a community context of the conservation with climate change, not just focusing on the foundation common species. So just because the plant's there doesn't mean it's going to support hardly anything, except decomposers. Okay, so this gets you, so in this third part, I wanted to show how the community effects and the mismatch that you would get with increasing climate change, which is also, the greater the mismatch, the greater the problems that it creates. Okay, so now I want to focus on solutions, because there are real solutions here, but they're up to a point. You change the environment so much, then the plants, you exceed the genetic physiological tolerances, and those plants are just simply going to die out. Okay, so the solutions are rapid evolution, acquire better mutualists, there's one, acquire better mutualists, you can move or you can die. Okay, and once you exceed the limits of the species, you're into the die zone, and I think that's going to be a pretty common area actually, depending on the rates of change. But these other things represent opportunities for us to help mitigate the problems. And the heart of all of these solutions has in part or large part a genetic component. And so we need to understand the genetic components of your target species. And so this looks at, this is a study that by Hillary Cooper et al. That will come out in a few weeks in Global Change Biology. And this looks at two different environments. So this could be low elevation and high elevation. It could be high stress environment, low stress environment. Take your pick. And this looks at trait values. And the trait value could be phenology, could be productivity, any of the functional traits that you identify as being important in your particular plant. And then here we see 4 different genotypes in one environment and the same 4 genotypes in the other environment. And these lines represent reaction norms. And this is, again, a really important thing for geneticists to understand these reaction norms, how phenotype will change as a function of environment. And so here we see variation from one genotype to another in one environment. And this is the genetic component of that population in that environment. And then in this other environment, we see the same 4 genotypes, and the variation between them is also the genetic component in another environment. The difference in the mean population response here and the mean population response here is the environmental component, and that is typically referred to as plasticity. Most ecologists think of plasticity as having no genetic component. It's just the environment. But that's not true. There's a genetic component to plasticity that can be quantified, and that's shown here in the G by E interaction. And so the slope of this line for this genotype in 2 different environments is steep. The slope of this genotype across 2 environments, this reaction norm, is shallow. And if they're statistically significantly different, you can then say that there's a G by E interaction and there's a genetic component to plasticity. Okay, so from natural selection point of view, or artificial selection that a plant breeder might work on, or an animal breeder for that matter, they can select for the genetic component here, and they can select for the genetic component to plasticity. This is not something that evolution works on here, or the plant breeder, because this isn't genetically based, so you can't select for it. So you need to partition the variance, and the only way you can really calculate this or quantify it, partition it, is through these common garden field trials. This is what agronomists, agriculturalists do in the Midwest. It is the heart of agriculture. And it needs to be the heart, my bias, of climate change research, because this is the genetics of it, and this provides ways to deal with it. Okay, so let's now talk about some of these solutions. So rapid evolution. We often think of trees as being very slow to evolve. They're long-lived organisms, and so they're not going to evolve rapidly. That's not true. A lot depends on the standing genetic variation within a population. And so as a result of the 2002 record drought here in northern Arizona, we had identified drought-susceptible pinyon pines, and in 1 year, after a record 1-year drought, nearly 70% of the drought-susceptible trees died. And growing adjacent to them were drought-resistant pinyons, and only about 20% of them died. And so in just 1 year, we could show that the genetic composition of the tree population shifted from a mix of resistant and susceptible genotypes to a mix that was dominated by resistant, drought-tolerant genotypes. Okay, so that change in gene frequency, that's the definition of evolution. And so we could show here on the landscape how climate change is an ecological event and an evolutionary event. Okay, so it's more complex than understanding just the genetics of the tree, because the tree has mutualists, such as mycorrhizae, that live on the roots of the tree. And so in a pinyon common garden, here is a seedling from a drought-intolerant tree. Here's a seedling from a drought-tolerant tree that were all planted at the same time. And so you can see the drought-tolerant seedling is growing about 2 to 3 times faster. And here, we looked at the mycorrhizal community. This is another ordination, a study that just came out in PNAS by Kitty Gearing and collaborators. And this looks at the mycorrhizal community of drought-tolerant adults and seedlings. And so you can see they're very similar. Then if you look at the mycorrhizal community of drought-tolerant, intolerant mothers and their seedlings, you see they're very similar. And the mycorrhizal communities of these 2 groups are very different. And so this starts to suggest that the genetics of the tree is not only important in affecting its innate drought tolerance, but it affects the mycorrhizal communities, which can confer additional drought tolerance or intolerance. And here in the bottom graph, we look at an experiment in which they're inoculated with different community types. And so if you inoculate a seedling, a drought-intolerant seedling with its mycorrhizal community, you can see that a seedling with mycorrhizae does much better than a seedling without those mycorrhizae. So the mycorrhizae clearly benefit these seedlings, but they don't benefit the intolerant seedlings nearly as much as the drought-tolerant seedlings. Drought-tolerant seedlings with their native mycorrhizae do much better than seedlings without those mycorrhizae, and they proportionally do a lot better than these. And so who lives and who dies is not only determined by the genetics of the tree, but the genetics-based community of mycorrhizae that naturally colonize those trees. So mycorrhizae, it represents a solution here. Inoculating with these type of mycorrhizae can make a big difference. Okay, so assisted migration. Controversial issue. I think we're going to be forced into it simply because projections in climate change, we're going to exceed the tolerance of a lot of species. And so you're going to have a tough decision to make. Would you rather be a purist and not have that tree there? Or would you have one that's the same species but from someplace else? And so assisted migration, I think, is going to become increasingly important. And assisted migration has been going on for thousands of years, and nurserymen have been doing this. The nurserymen move everything everywhere, in many ways the scourge of environmentalists because of moving everything around. And I can bitch about this because I grew up in the nursery business, and so I know exactly what nurserymen do. And so here's an example of digging a tree up and moving it. We move trees from hundreds of miles to thousands of miles. And so they cause problems, but we can get a lot smarter about it. And so if we take information on assisted migration, we can improve a situation that could get even worse. And so let's go back to the performance data based out of a common garden. Okay, so here we are at a cool garden, high elevation, short growing season of Fremont cottonwood. And so I showed you earlier that the locals did best, but then if the climate changes 2 degrees, 4 degrees, 6 degrees, by sliding down this scale to plants that are growing in hotter and hotter environments, we can find the individual populations that are already genetically adapted to a 2 degree change, 4 degrees hotter, 6 degrees hotter. And so we can select those populations that are best suited to the high elevation environment, say 20 years from now, 40 years from now, or over whatever the climate change projections suggest. Same thing for the intermediate population. We know where we can predict where we need to get the populations for the future. But at the lower elevation, at near sea level, there's no lower population to go to. And so those populations are at the trailing edge of the species distribution, and they're likely to go extinct because we can't find ones that are already genetically adapted to more severe environments. If it's a really important species, we can genetically engineer it. We can do gene stacking. There are lots of solutions. Right now, politically, we couldn't do it. But if things get bad enough, we're already starting to do this with American chestnut. I think we're going to go in this direction, just my gut response, but American chestnut will be the first test case, and it's happening right now. Okay, so phenotypic plasticity. Here's an example of the environment. There's no genetic component in this particular graph. There is, if I included other data, but this is just one tree genotype, and it looks at leaf morphology. And so here we see 5 leaves from the same tree. And this is at their home site in Keeps Canyon, Arizona, among the highest elevations that you will find Fremont cottonwood. Okay, so if we propagate these trees from high elevation and we move them down to a lower elevation garden at Chevron, where the temperature is 3.2 degrees centigrade hotter than their home site, look at what happens to the leaves. The leaves are about 1/3 the size of the home site. And so the first response to climate change in this species is a reduction in leaf size. Okay, and there's a lot of other things, including stomatal density and other things that go along with this. And notice that the leaves are still quite symmetrical. Okay, so then we take trees from up here and we move them down to the lowest elevation site the Fremont cottonwood naturally occurs. And you can see down here, leaf asymmetry is all gone. I wouldn't even recognize that as a Fremont cottonwood if you gave me the leaf. And the leaves are very asymmetrical, and as asymmetry goes up, productivity goes down, mortality goes up. And so there are very strong ecological correlates here. But if this sort of thing is generally true, you can walk out to any tree population and start looking for diminishing leaf size and diminishing symmetry, and you can start to identify populations at risk and start to do something about it before they actually all die like they are in the Bill Williams River. So I think this is a very important biomarker using plasticity as a trait for what bad things that might happen. Okay, so the next question I want to ask is, as you do assisted migration, you can deal with the tree, but you haven't answered the question about the community that it supports. And so, like in the case of Bill Williams River, if we had propagated those trees and moved them someplace, would we effectively move the communities that they supported? Would they find it? And so here's asking the question, if you build it, will they come? And so the answer to this question, this is the first study that I've seen on this, and it's not published yet. It's some work by Art Keith. And so the answer to the question is, if you build it up to a point, they will come. And so here we have a common garden, and the garden here is all the same garden. And then we move plants from 18 kilometers away, nearly 50 kilometers away, nearly 100 kilometers away, into the same garden. And then we looked at the communities that these trees supported in the wild and the communities they supported in the garden. And so with an 18-kilometer transfer distance, there's significant differences, but they're not huge. At nearly 50 kilometers, there's still significant differences, but the differences have increased. But again, they're not huge. But once you get up to nearly 100 kilometers, the differences have now become really great. And so there's some point at which if you move it, they will come, but this is probably species specific and it needs to be defined for each of those species. And so we need a lot more of these tests. Okay, so in summary, what this shows, number one, plants are very locally adapted, and climate change effects on locally adapted plants will be great. It's a negative effect. Plant genotype can define communities, and so if climate change affects those genotypes, then negative effects on the communities. Climate change, third part, climate change creates a mismatch of locally adapted becoming locally maladapted. And again, climate change is going to have a big effect there. In terms of the solutions, there are passive solutions that don't require us to do anything. The passive solutions include things like the standing genetic variation that is already at the site, phenotypic plasticity that moves you in the right direction, and then again cut off the human intervention. So once you exceed the genetic variation of the local population and plasticity, then you're into assisted migration, genetics-based ecosystem engineering, and then true genetic modifications like true genetic engineering, not the genetic engineering of moving things around, different genotypes, moving different genotypes around. And so I think we need to discover all of this stuff for especially foundation species, species that are most biologists, ecologists would agree are incredibly important species. I mean, if we lose the ponderosa pine forest here, it will fundamentally change everything. And so ponderosa is a foundation species. But how many foundation species are there? How many foundation interactions are there with a bird or a mammal? I mean, beaver are clearly foundation species of cottonwood. And so we need, in each ecosystem, we need ecologists and biologists to say, who are the foundation species? Because they are, by definition, community and ecosystem drivers. I think a lot of species are just tag along. Those interactions of foundation species create the environment for other species to exist. And everything we've done would argue that there's just a handful of what people would generally recognize as clear foundation species. Okay, I'd like to end by recognizing a large group of collaborators. This is our current group of collaborators. NSF has been very generous in their support over many years for this. These are other university institutions that have collaborated. And then, perhaps most importantly, here are the land management groups that have allowed us to use their lands. And I want to emphasize this incredibly. You can't get these people involved soon enough. Because my experience tells me, unless the managers are involved in the project, I don't care how good the science is, they will never incorporate your science. And by having them involved at the get-go, they are far more likely to get on board and use your findings in land management practices. Thanks very much.